I am a medical oncologist. I'll just give you a few words about how I get to where I am in what I'm doing at Dana-Farber right now. I started out, I did my fellowship at Dana-Farber many years ago and worked in a lab, actually working on the signal transduction, and I'll explain that. Signal transduction is exactly what Lou was talking about in terms of these pathways that exist in cancer cells, but I actually worked on them in leukemia. I missed patients a lot, so I ended up at that time in the mid-1990s or so working and seeing patients in our breast cancer clinic and then started seeing patients with gynecologic malignancies. And as I took care of more patients with gynecologic cancers, I realized that there was definitely a void in the research that was going on. So about 8 years ago, I became the clinical director of our GYN program at Dana-Farber. And my focus is predominantly on ovarian cancer, endometrial cancer, and cervix cancer. The ties to the BCRF, and again, I have to thank the BCRF very much because of their intelligence and their foresight into understanding that there are ties between ovarian cancer and breast cancer. So what I work on specifically, and as Lou and everyone else here has mentioned, we work as teams, and especially in Boston, we collaborate a lot. But I work on a project that looks at the genetic similarities between a specific type of breast cancer called triple-negative breast cancer and a type of ovarian cancer called high-grade serous ovarian cancer. So just a few words about ovary cancer. We split it into 4 groups right now. Serous tumors, endometrioid, clear cell, and mucinous. And that's based upon what the pathologist sees underneath the microscope. We're not as smart as our breast cancer colleagues where they have actually identified molecular targets like HER2 or even ER. But we've always known that these 4 distinct subtypes behave clinically very differently. So for example, serous tumors or high-grade serous, and that's spelled S-E-R-O-U-S, are very sensitive to a drug called platinum, whereas a type of tumor, mucinous tumors, typically are not sensitive at all to chemotherapy. And what's occurred is that these histologic or pathologic subtype, in other words, what the pathologist sees under the microscope, we can actually differentiate from a molecular viewpoint. And that's what we've learned in the past few years, which has actually been very exciting. So how that ties to breast cancer is that the serous tumors are sensitive to chemotherapy, such as carboplatin or cisplatin, and the BRCA gene, breast cancer gene, is frequently either mutated, meaning changed, or somehow different, so it's not working. Triple-negative breast cancer, and Judy Garber here, who has done a lot of work on triple-negative breast cancer in terms of the sensitivity of triple-negative breast cancer to platinum, to cisplatin specifically. She's published papers on that. And that's also the type of breast cancer that is associated with a BRCA mutation, typically the kind that's inherited between families. So our project intertwines and looks at the similarities between the two, and Charles Wang, who's sitting all the way in the back. Charles, can you wave your hand? There he goes. So Charles does all the work. So really Charles should be up here. But we have found, Charles has found, that there are similarities from a molecular standpoint in that the genetic piece of these cancers is actually very unstable. And the other observation that we've been making are these drugs called PARP inhibitors, P-A-R-P. PARP is an enzyme, you've heard about enzymes, that basically is like the ambulance that drives to a piece of DNA that's damaged, and it brings a whole bunch of friends with it as well to help repair it. PARP inhibitors have been in the news a lot. They have come in and out of favor for breast cancer, specifically triple-negative breast cancer. We do know that they work very well in high-grade serous ovarian cancer, and in fact, that's one of the more exciting drugs that we've been testing. But for some reason, they may not work as well, but they certainly probably work in certain subsets of triple-negative breast cancer. So what we're doing now, this next year, is looking at patients' samples of either triple-negative breast cancer or ovarian cancer. These patients have received PARP inhibitors, and we're going to look at the genetics of these tumors and see if the patients actually responded to PARP inhibitors or not, and try to figure out, maybe specifically for breast cancer, how we can make those cancers maybe more sensitive to PARP inhibitors, or figure out why they may or may not be sensitive, maybe pull out a subgroup of women who might be sensitive to them. So that's my and our project.
That's great. Thank you very much.